Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. My goal here is to find the exceptional folks in every field and ask them questions and hopefully get them to say, that's a good question, because it means I've gotten to some information they have in their brains that maybe they haven't spoken about before. Today I have Phillip Zamore. He's the chair and professor at University of Massachusetts Medical School. We're going to be talking about a big-time landmark FDA approval that allows gene silencing. It's a new method that he's been working on. So, Phillip, thanks for coming.
You're welcome.
Yeah, what is gene silencing, first of all?
Gene silencing, or as it's often called, RNA interference, or RNAi, is a way of destroying messenger RNAs from a gene so that the protein product can't be made. So in its simplest form, it's a kind of off switch that you can add to cells to turn off a disease gene, for example. And unlike other mechanisms that attempt to do that same thing, it acts using a natural cellular pathway, which is why it works so well.
Where does it halt the process? Is it a gene is transcribed, but then it cannot be made into the constituent protein, or does it prevent like epigenetic marks? Let's say a gene being looked at and transcribed in the first place.
No, so the gene's transcribed, but then something called a small interfering RNA programs a natural enzyme called Argonaute-2 or AGO2, AGO, to actually make a single scissors-like cut in the messenger RNA, which ultimately leads to its destruction. But even that one cut prevents the protein from being made.
So what happens to the transcribed gene? Where does it go? Does it get absorbed by the cell, or does it migrate to somewhere, or what happens?
No, it gets broken down into its individual nucleotide parts and just recycled by the cell. Basically, the process of destroying it is entirely natural. The only thing that is new is the sequence of the siRNA targets an RNA that normally would be stable to be destroyed by the natural pathway.
So I've heard of a lot of places doing gene knockouts. Is this perhaps a superior method to doing a gene knockout, because you could leave the genome intact but still silence the genes you want to silence?
Yeah, so I mean, I think you can divide the world's diseases into two broad categories. One is an inborn error where there's a mutation in the genome and you want to turn off the gene forever. And then the other is where you want to, for example, lower the amount of gene product instead of turning it off completely. So a knockout can only do the first kind. And the other thing about a knockout is if it has side effects, you can't reverse them. So once you've done a knockout, you can never go back, which makes it, I think, a really good tool for studying model organisms in the lab, but maybe not yet mature enough for use in humans. Whereas RNAi is like a drug. If you stop taking it, you stop its effects.
Yeah, that's really interesting. So this could be cell type specific, it's dose specific, it has a timeline, it could probably be time release. I mean, you can modulate a lot of effects with it.
That's exactly right. And the drugs, there are two drugs on the market now that use this mechanism, two siRNA drugs. And what's common in both cases is that the product that the gene product, the protein that causes the disease, is made in the liver. So right now, you can direct siRNAs to the liver right now very efficiently. that technology has really become a mature platform, so that once you pick a disease gene that's made by the liver that you want to turn down or almost off, there's a clear drug development path to do that. For places in the body other than the liver, the technology is still being developed. I think the next one you'll see is probably in the central nervous system. Maybe after that, you might see the technology being used actually in the placenta for diseases like preeclampsia. So the two sides of the drug development strategy are, first of all, to make the drug safe and effective, but also make it get to the right tissue. And right now, the only commercial drugs that are based on siRNAs are those that go to the liver and turn off a gene product production there.
Why is the liver particularly amenable to these kind of drugs?
I think for two reasons. The first is just historical, in that strategies for delivering any kind of oligonucleotide to the liver have been worked on the longest, and so sort of luck was on the side of getting that to work well first. But the second is there's a lot of blood flow through the liver, so you know that by putting the drug into a human being either under the skin, which is called subcutaneously, or into the bloodstream by infusion, that it'll go to the liver. So the question then is, having gone to the liver and the blood, will it get taken up by the hepatocytes? And the trick there is specific sugars have actually been attached to the siRNA to enable them to bind a receptor on the surface of hepatocytes.
So, all right, how would this be tailored then to other cell types? Like, what's the important criteria that this drug will go into the right cells, the right cell type? How do you account for, I guess, the membrane particulars, or how do you know how this will work?
So what people have been looking for are ligands, like the sugars that are used to deliver to the liver, that they can attach to siRNAs that'll cause them to accumulate in other tissues. And typically those strategies involve a combination of trying different ligands that are known to bind to proteins that have been well studied, that are on the surfaces of specific cell types, and a lot of luck. For a long time, this GalNAc technology, this sugar that sends it to the liver, didn't seem like it was working, and ultimately it was a matter of tweaking it, and then it became very effective.
So how did you discover this in the first place? What was it? How did you get this idea, and how did you figure this out?
Yeah, so I'm a biologist, and I have not really ever set out to develop technology per se, or drug therapies, but like many biologists of my generation, my eyes are open, and I'm looking to see what discoveries we make about nature might have practical use. So back in 1999, 2000, my colleagues and I, Tom Tuschl, Dave Bartel, and Phil Sharp, started to study a process called RNAi that had been described in the nematode C. elegans. And when we figured out how to study it biochemically in fruit flies, we discovered that the sort of active ingredient in the pathway were these little tiny RNAs called small interfering RNAs, or siRNAs. And we thought, well, if these are found in nematodes, and that we knew at the time they were found in plants, and they're found in fruit flies, we thought, because of the way evolution works, they had to be present in humans doing the same thing. And so we got the idea to start a company, Alnylam, that would try to use our discoveries in fruit flies initially to develop drugs for use in humans based on the same mechanism. And along that path, while we were starting the company, Tom Tuschl's lab, he had then moved to Germany, discovered that you could actually make these siRNAs in the lab and get them into cells and culture. So between those two discoveries, the one in fruit flies and the one in immortal cultured human cells in a dish, it looked pretty promising. And so we were young and naive and a little bit arrogant. And we started this company, and we thought it'll just be a couple of years. We'll get the company will get into work in humans. Well, it took 14 years. The journey, I think, was very similar to other drugs that were based on biological discoveries, just about the same time scale, but we were young enough that we thought it would be different for us, and it wasn't.
So at what point would gene knockouts be a thing of the past, and this would be the preferred method of experimentation?
Could you repeat that?
At what point do you think that this will be robust enough where gene knockouts will be a thing of the past, and this will be the preferred method instead?
No, I don't think this will ever replace gene knockouts. That in terms of experiments rather than therapies, they serve very different purposes. So there are two kinds of gene knockouts: regular knockouts and conditional knockouts. So a regular knockout removes the gene from every cell in the body. Often that is lethal, and so you can't really study it. Conditional knockouts allow you to remove the gene from a particular cell lineage. Often that allows you to study the function of the gene, but more often than not, again, removing the gene completely can kill the cells that you want to study. And so at that point, people turn to RNAi, and instead of completely removing the gene, they reduce the amount of the gene product by enough to see its effect, but without killing the cells. So from an experimental standpoint, they're extremely complementary techniques.
But why would this not be able to replace it? It seems like the dials on this are a lot more fine than the dials on any type of gene knockout.
Well, first of all, I would say because often you find the opposite case where reducing the level to small amounts still is enough for the cell to be fine, in which case a knockout is necessary. And also because knockouts versus RNAi, the difference tells you something. So in general, you like to do both. One is what happens is answers the question, what happens if you didn't have this protein at all? And the other is what happens if you don't have enough of the protein? So, for example, there are cancers where tumor suppressors simply are not made at all. So if you want to understand those, you need to knock out the tumor suppressor.
Okay. I just didn't know if you can get it to such a low level that it approximates a knockout, and therefore you wouldn't need a knockout.
There are cases where that's possible, but in general, if you can get it below, say, 10 or 20% of the original level, you're doing really well. And
are there methods to upregulate genes selectively, or is this a method now where you can do that where you couldn't before?
No, I would say that you can't directly turn a gene up using this strategy, but because genes are part of larger regulatory networks, you can often target a repressor of a gene, which will allow the downstream gene to have increased expression. And similarly, Alnylam has been developing siRNA therapies for hemophilia in which an inhibitor of clotting is actually reduced, allowing the clotting pathway to be more sensitive and regain function.
Does this appear to affect any methylation or epigenetic marks?
So far in mammals, no.
That's too bad.
Well, it depends.
Well, maybe.
It depends on your perspective. If you're developing a drug and it does that, it actually comes under a much higher level of regulatory scrutiny, and
Really?
it needs a much more demanding safety study. Anything that changes something in the DNA or chromatin it gets treated very differently from something that acts just in the cytoplasm to chew up an RNA.
Oh, is there a name for those different designations?
Yes.
Are there any drugs that do appear to remove epigenetic marks?
Yeah, I mean, there are HDAC inhibitors, for example, that do that. But generally, those are anti-cancer drugs. People have certainly tried drugs like that historically for sickle cell anemia to try to remove the repressive methyl marks from the DNA of the fetal hemoglobin gene. It hasn't been, I think, a very effective strategy. And I think if you were to develop something like that today, the level of regulatory scrutiny would be pretty high to make sure that it was doing that only in erythrocytes and not in other cells.
Do you think that this could be used to prevent bacteria from infecting us, or to prevent, as an antiviral somehow? Or if a cell gets infected by a virus, could it be a way to block the action or the construction of more virions?
So to take your first question, can it be used as an antibacterial? Probably not. As an antiviral, absolutely. And I have a colleague who right now is working literally round the clock with her lab to try to develop siRNA antivirals for COVID-19. And if you were a member of the majority of animals on the planet instead of a member of this tiny little minority of mammals, you would routinely use siRNAs as an antiviral. So in arthropods and nematodes, the siRNA pathway is one of the main defenses against viruses. So every beetle,
that's a good sign.
every butterfly, every fruit fly, they're all using this pathway to fight viruses.
Where does their siRNA material come from? I guess it must be made endogenously, obviously, but where does it come from?
Right. So in terms of viruses, many viruses are made of RNA. So they store their genetic material in RNA, sometimes double-stranded RNA, sometimes single-stranded RNA. But even when they're single-stranded, they have to go through a double-stranded intermediate. And naturally, cells chop up double-stranded RNA to make siRNA.
So in a beetle, it gets infected by a virus. What material is the beetle using? Is that known to create these siRNAs to block the viral action?
Yeah, it actually uses an enzyme called dicer that chops up the double-stranded RNA genome of the virus and directly makes the siRNAs that way. So in a way, if you think about the parallel to our antibody immune system, the big trick in antibodies and in T-cell receptors is that the presence of something like a protein, for example, on the surface of a pathogen or made by a virus, triggers a specific immune response against that exact protein, right? That's the basis of vaccines, to basically produce a lasting memory of how to make antibodies for the rest of your life against that feature of the pathogen. So the RNAi system is analogous in that it takes the genetic material of a virus and uses it directly to make siRNA. So when the virus isn't there, it doesn't make the siRNAs. And when the virus gets there, it recognizes the fact that it's double-stranded RNA, which is something unusual in a cell, and it chops it up into siRNAs that then can be used to destroy the messenger RNAs that the virus makes and the genomic RNA.
Huh. That's really interesting. That's amazing. I wonder why our cells don't do that. Or maybe they do, just not strongly enough. Or
so there are sort of two main pathways that accomplish these conversions of double-stranded RNA into smaller RNAs. One is called the microRNA pathway, and one is called the siRNA pathway, and they both use versions of the enzyme dicer. In the antiviral RNAi pathway, dicer is extremely efficient. And in the microRNA pathway, it's a much more tame enzyme. And so in mammals, we use this sort of wimpier version of dicer to make small RNAs called microRNAs that regulate our own genes, but we don't normally use it as an antiviral. Now we can take advantage of that pathway with synthetic siRNA drugs, but the antiviral pathway using this really efficient version of dicer was lost somewhere on the way to the lineage of mammals. but it was retained in arthropods, so lobsters and flies and beetles.
Interesting. Are we able to look for the presence of even small amounts of this sRNA in our own cells when we get a viral infection? Do you think it's there at all, or it's not there at all?
I don't think it's there at all.
Interesting.
But viruses do actually exploit that to make their own microRNA.
Oh, tell me a little bit about that.
So in addition to all the microRNAs we make to normally regulate our genes, there are a few viruses like hepatitis B virus that actually make their own microRNAs that turn off our mRNAs in order to allow the virus to grow. So the virus actually exploits our own pathway to turn back on us. So kind of the opposite of what the RNAi pathway is doing normally.
You mean is the virus starting up this pathway somehow to block the production of some of the proteins that we would need, or ones that would be antithetical to the virus?
Yes, exactly.
Huh, that's crazy. So what is the origin of the creation of these sRNAs? You said in arthropods, it's there, but the cell recognizes this this double-stranded RNA. But I don't know. Is there more known about the recognition of it, and literally what the start of this whole process is, and what are the substances that literally start the process?
Yeah,
Creation of the sRNA itself.
So the last common ancestor of plants, animals, and fungi had this pathway and used double-stranded RNA generated either by viruses or by the cell itself to make siRNAs. And then in plants, it was retained as an antiviral. And then in animals, many of the lineages, like arthropods and nematodes, retained it. But on the way to mammals, it was lost. And the idea of using double-stranded RNA as the signal to make siRNAs is really clever. And it was Craig Mello and Andy Fire who won the Nobel Prize for the discovery of double-stranded RNA as the trigger of RNAi. And by using double-stranded RNA, the cell can separate normal cellular RNA, which is very rarely double-stranded, from viral RNA. But it can also specifically make double-stranded RNAs that will be recognized in a similar way and either make siRNAs or microRNAs that can do cellular function. So in plants, siRNAs actually can direct epigenetic marks that regulate genes in the nucleus. Same in fungi. In animals, there are very few examples of that because there are other pathways that have replaced that RNA-guided strategy.
Okay.
There's one related pathway that my lab has worked on for a long time called the PIRNA pathway that uses many of the same proteins but doesn't begin with double-stranded RNA that actually can change epigenetic marks, including DNA methylation in mammals and chromatin methylation in arthropods. And that pathway protects our germ cells from transposons.
Can we tell, for instance, when someone has a viral infection, like for instance now with COVID, whether the virus is activating these sRNA type elements, and how would we use that information if we did know to counteract it?
Yeah, I mean, my bet is they're not because of the way mammalian biology works. But if they were, the way to do it is to get samples of the infected cells, and we use a technique called high throughput sequencing to specifically sequence millions upon millions of the small RNAs in the cell. And that will reveal whether or not any are being made from the virus.
Okay. So what do you think is going to be the, back to the technology, what's going to be the near-term future in terms of ability? You said in the liver, this seems to work pretty well, small molecule drugs that can accomplish this RNA interference. What's next, or what's coming soon?
Yeah, so I think the most exciting thing that's just on the horizon, I'm not quite sure how the pandemic has slowed it down, if at all, but before the pandemic, I would have predicted you'd see approval this year, is of an siRNA drug that blocks the production of a protein in the cholesterol biosynthesis pathway, and is likely to be a replacement for statin. And the two really exciting things about that drug are that it appears to have, at least in clinical trials, which were done with thousands of people, a much more favorable side effect profile. So it has fewer side effects. And the way the siRNA drug was designed, it's incredibly stable. And so you would only need to receive it twice a year, which is a huge advantage for people with high cholesterol, because forgetting to take a daily medicine is one of the main reasons why statins don't work as well as people had hoped.
Oh, wow.
So if you could just go to CVS and they would give you an injection that felt not so terribly different from a flu shot twice a year, I think, people's cholesterol would be under much better control. So that would be different. I think that's the near term. I think the long term is to develop siRNAs that can be injected into the nervous system, maybe twice a year, that would allow you to treat diseases like Huntington's disease. And there's a lot of work being done both in companies and in academic research labs to try to bring that about.
Okay. Well, very good. Phillip, what's the best way for people to find out more about this phenomenon and to read some of the work that you've put out there?
Well, all of the work that I've done is available at either my Google Scholar homepage. You just search for me by my first initial and my last name. Nature has had over the years a number of really great articles about the field. And I think they now have a permanent set of pieces for the sort of scientifically literate layperson on their website. And if you want to know more about drug development in the field, going to any of the websites of the companies that try to develop these compounds like Alnylam or Dicerna, there's often a ton of information for patients.
Okay. Well, very good. So those, any other ways to follow up, or those are the best?
I think, truthfully, Google is not a bad way. You'll get if you just search siRNA, you will get a lot of information, and you just look for reputable sources like universities, websites, or the big scientific magazines, Scientific American, things like that.
All right, very good. Well, Phillip, thank you for coming, and it's really interesting work that you're doing.
Thank you. It's a pleasure.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.